economics. So why development
First,
is required? If you have got, for example, antibiotic producing strain of penicillin, Penicillium notatum fungus, and we want to screen for maximum probability, na? If we are doing some mutation combination, we are getting 10,000 spores. आपण कुठला एक्सपेरिमेंट लावला आणि त्यामध्ये काय केलं आपल्याला, ड्युरिंग दिस एक्सपेरिमेंट वी गेट 10,000 spores of this penicillium. There is only there will be few clones which are giving exceeding result or maximum productivity. How they will calculate the productivity? Per unit glucose, how much units of enzyme or active molecule they are producing. So here screening is required. No? Whenever there is development of a new variety, sugarcane, if you are studying sugarcane, uh, very nice variety is CO86032, 86032. So it is very popular. And other one is why it is popular, because it has got highest sugar recovery. It exceeds about 12. And second is CO265. Why this is famous? Because it has got low recovery but highest productivity. For farmers, CO265 is very good, but for industry, CO86032 is very nice because they want productivity. So how they produce this? They are getting during the breeding program, they will get thousands of clones, and out of this only few they have to select. So screening is very important. Speedy the screening. And now you are seeing the COVID situation. Now in whole worldwide, lot many vaccines are coming, but out of this, which is effective? We have to do the screening work, and it should be done how? Very fastly, because now the pandemic has extended worldwide, and lot of patients are falling prey for this COVID-19. So there is urgent need for this vaccine. So how can we ease this screening process? By using these computational techniques. Well,
sir.
now what we can use more? You can use robots, data management, softwares, different array systems, and we will ease the screening program. So this is very important step during development of a product or a process, the screening step. Now, for example, we are producing enzyme activity. We want highest enzyme producing organisms. What we do? Screening is done in 250 ml flask. आणि जेव्हा we are standardizing this in 250 ml, then we are going for scale up प्रोसेसच्या बाबतीमध्ये. For example, enzyme extract करायचं तुम्हाला, or you want to produce ethanol. So first you will do in a small capacity, and then you will go for larger capacity. Then next step is search strategy in traditional biology and bioinformatics. Now, what strategy you should use? Biology is there, then traditional biology is there, and bioinformatics is there. So as we'll study, these are all interrelated. How? Naturally आपण काय बघतोय? For example, we want a antivenom produced from snake. What we'll do? We'll study the biology. How we'll study the biology? For example, the wild animals are there, monkey is there, and other is there. If they are getting snake bite, if they are getting snake bite, what they will do? They will know the remedy, what plant or what root they have to eat to get protection from this venom. So they will go and they will uh, go in the wild, and they will select that required plant which has got anti-venom activity, and they will consume that plant. So in this wild area, the Adivasi people are there. What they will do? They will observe this traditional knowledge, and they will apply for what plant is there, which plant part it has uh, consumed to give protection, and they will take this knowledge and they will keep it and they will try it on humans whenever in that area anyone is getting affected through this snake venom, then they will use that plant. Similarly, आपल्याकडे देवगिरीज होते माहिती ऐकले देवगिरी फॉरेस्ट ऐकलंय का देवगिरी.
First,
So outside of the every village there was a sacred forest. This sacred forest contains lot of medicinal plants. So Ajivaisa Batwa, it is famous. So what happens? The elderly people, they were knowing what plant extract you have to use for what disease. For example, you have got cough, you have got fever, you have got dengue, you have got any malaria or any other symptoms, heart problems. So all naturally This medicine was available in that area itself. So every area has got different. If you consider in India in Maharashtra we have got different plants. In Konkan region we will get different plants. In Sahyadri Western Ghats we will get different plants, and again in this drier area we will get different plants. So everywhere for cough is everywhere, but remedy plant is different. So here what is required? For example, if you are working in healthcare, what we have to do? We have to collect the specimens. We have to observe the systems. Then we have to do data collection. How you will do data collection? You will go to these people. You will meet these people, and then what you will do? You will document the photographs of these plants, their process, and you will complete a database. Then what you will do? After that, what you will do? You will take that plant. You will do its tissue culture. You will extract. You will look for the which active ingredient is present in that. You will separate it, and for separate after separation, you will go for commercialization. What Patanjali did, Baba Ramdev, everyone has heard, no? Patanjali. They started their
Yes.
on cycles and they were distributing these medicines. They're collecting the knowledge from the local people in Himalaya. Now they have established their brand. So this process can be eased by using computer applications, traditional biology, applied biotechnology, and the search strategy is very important. If you are having, for example, if you are studying fever or cough, and there are 15 plants, so which plant is effective? What plant combination is effective? Which is easy to grow? All these parts you have to study, and then you can bring Ayurvedic cough syrup in the market. So screening strategy is very important, and how bioinformatics is used here. Then next is aims of biotechnology. Now advances made in the past decades. कुठले कुठले आहेत ऑर्गॅनिक केमिस्ट्रीमध्ये. ऑर्गॅनिक केमिस्ट्रीमध्ये काय केलं? व्हॉट दे डीड? ऑर्गॅनिक केमिस्ट्री आर्टिफिशियल सिंथेसिस ऑफ द केमिकल्स, स्मॉल केमिकल्स और एक्सट्रॅक्शन, त्याच्यामध्ये खूप मोठा रिसर्च झाला. देन कॉम्बिनेशनल बायोसिंथेसिस, देन मेटाबॉलिक पाथवे इल्युसिडेशन. कॉम्बिनेशनल बायोसिंथेसिस म्हणजे काय? फॉर एक्झाम्पल, what by which biosynthesis the insulin molecule is synthesized, or antibiotic is synthesized, amino acid is synthesized, vitamin is synthesized. The metabolic pathway they studied all the metabolic pathway. म्हणजे काय केलं? फॉर एक्झाम्पल, इफ यू गॉट क्रेब सायकल प्लांट मध्ये होती, अॅनिमल्स मध्ये होती. कुठे होती क्रेब सायकल? कॅन एनिवन टेल? क्रेब सायकल कुठे होती?
हॅलो.
हा, क्रेब सायकल वेर इट इज फाउंड?
प्लांट मध्ये.
हा. So there are multiple enzymes, multiple products are developed. So this is study of metabolic pathway. It is very short cycle as compared to other cycles. For example, they want to synthesize specific vitamin. So vitamin they will require amino acid, then they will require inorganic metals, and they will get combined by using n number of enzymes, and you will get final your vitamin. So these metabolic pathways they have studied. Now metabolic pathways, they how they will be governed? They will be upregulated. Last time I have given an example of starch is present in the environment, and you want to the bacteria want to utilize the starch. So it will secrete amylase. How it will secrete amylase? It will induce the gene. Gene ला induce केला म्हणजे काय होईल? Promoter will act, and the gene will get induced. Over expression of mRNA will take place, and this proteins peptide chains will form, and they will give active amylase enzyme, and this enzyme is secreted outside the cell. Then whatever starch is there, it will get degraded and the glucose will be absorbed. So it comes to gene shuffling, then direct evolution of proteins. So all this data we have got, how genes are regulating the production of different enzymes, how they will govern the metabolic pathway, then how organic chemistry is used to extract this molecule, then what it will cause. We can use this strategy in traditional biology. Then we will take this to bioinformatics, and by using these tools, we can get lot of new products. and time period is very important. Time period is very important. Why it is important? In any this, why time period is very important?
सर मला ऐकू आलं नाही, पुन्हा एकदा सांगा की काय म्हटलं तुम्ही?
During the drug discovery, the time period is very important. Why?
हॅलो, कशासाठी?
ड्रग डिस्कवरी जर कुठली नवीन झाली तर टाइम पिरियड इज व्हेरी इम्पॉर्टंट. व्हाय? इट इज रिलेटेड टू पेटेंट. हाऊ?
To observe its results.
कारण का ड्रग ह्याच्या काय काय ज्या स्टेज असते त्या फॉलो करायला लागते त्या.
बरोबर. मग काय करायला पाहिजे की फॉर एक्झाम्पल ड्रग घेतलं नवीन आणि ड्रग त्यांनी काय केलं, दे हॅव डिस्कवर्ड अ न्यू ड्रग. ड्रग इज नथिंग बट ए केमिकल, काहीतरी स्ट्रक्चर असेल XYZ कंटेनिंग कार्बोहायड्रेट, प्रोटीन और एनी कोफॅक्टर. बरोबर ना? ड्रग काय वेगळं काय दुसरं त्यामध्ये सगळे ऑर्गॅनिक मॉलिक्युल्सच आहेत ना. पेनिसिलिन असेल, तुमचं स्ट्रेप्टोमायसिन असेल, सायक्लिक रिंग्स असतात. सो इफ यू गेट फॉर एक्झाम्पल ए न्यू ड्रग, सो दिस न्यू ड्रग यू हॅव discovered. Now you have to study the application of this new drug in, for example, say this COVID situation again. You want to study antiviral drug. So what you have to do? You will file a patent. The patent is protected for 15 years. So before this, in drug discovery, you have wasted 5 years. 5 years you will take for clinical trials, and you will get only 5 years to make profit. कळलं मी काय म्हणलं? Patent is protected for 15 years. Whenever you have discovered during discovery, you have lost 5 years. For clinical trials, you are lost more 5 years. Clinical trials म्हणजे काय करणार ते? What they will do? They will take this effect on the mice, or they will take rabbits, and they will study what they are getting side effects. Then they will go for humans. By using human consent, they will take the trials on humans, and then they will tell that this formulation is important, this concentration is used, and it has got this effect, and the way of the metabolic pathway, how it is activated. हे सगळ्यांमध्ये गेले 10 वर्ष 12 वर्ष. नाउ दे विल गेट ओनली 3 to 5 years.
हॅलो.
हा हॅलो.
हा सर, प्राजक्ता वेटिंग वर ऍड करताय तिला.
ओके. So what I was telling, that only you have got 5 years to book your profit. बरोबर ना? If you are taking drug discovery and all those process, if you are taking 10 years
ते
or
10 years गेले,
14
मग
years,
5 वर्ष
14 years,
फक्त.
you will get only 1 year. मग हा टाइम पिरियड कमी करायचं काम कोण करणार? बायोइन्फॉर्मॅटिक्स टूल्स. How
Yes.
they will do? Last time I have given an example. They will create a virtual lab. तुमचे virtual lab, virtual practical आता तुम्ही ऐकत असाल. And in this virtual lab, they will put the same size simulations as it is going in the cell. And they will put that drug inside that chemical structure, and they will do reaction at a time with carbohydrate, protein, fats, amino acid, cell membrane, and they will study the effect. Soft lab, मी सांगितलं 2 पद्धतीचा रिसर्च होतो. One is wet lab and one is soft lab. So they will use high capacity computers, good quality softwares, and they will employ this team to study 1,000 drugs and effect of it on 1,000 organisms. मग 1,000 into 1,000 तुम्हाला किती करायला लागले? ट्रायल करायला लागले असते 10,000 एक्सपेरिमेंट लावायला लागले असते. यू विल डू दिस एक्सपेरिमेंट इन द वन शॉट. तो तिथं तुम्ही 1 महिना जरी वाचवला, इन दॅट केस इफ यू save at least 1 month, you will get that much, you can book maximum profit. So these bioinformatics tools, databases, bioinformatics tools, they are getting wide range of applications. So this made why I'm preparing this PPT is why bioinformatics was evolved. बायोइन्फॉर्मॅटिक्सची गरज का लागली? तर ह्या सगळ्या गोष्टींमुळे बायोइन्फॉर्मॅटिक्सची गरज लागली. आलं लक्षात? हॅलो.
हा सर.
सगळ्यांना कळालं?
हो सर.
आवाज सगळ्यांना येतो ना व्यवस्थित?
हो.
आज हर्षदा जॉईन झाली नाही का? हॅलो.
नाही सर.
सो दिस इज प्री हिस्ट्री. आपलं लेक्चर कशावरती आहे? तर व्हॉट इज द history before bioinformatics was developed? What are the needs in these circumstances the bioinformatics field was developed? सगळे मॅथमॅटिशियन्स एकत्र आले, कॉम्प्युटरचे लोक एकत्र आले, बायोलॉजीच्या लोकांनी त्यांचा डाटा शेअर केला आणि त्यांनी हे सगळे तयार केले. वेगवेगळे सॉफ्टवेअर्स असतील, वेगवेगळे ऍप्लिकेशन्स असतील, टू इज द discovery of new molecules and applications, various applications. Now next slide is biotechnology and agriculture. Now everyone knows the global population is increasing, land is decreasing, people are suffering from poverty, so this economic slowdown, then there is no good quality of food is present, then malnutrition is taking place, and due to all this it is very very important to have advancement in agriculture and how you can use biotechnology to increase the crop yield. आलं लक्षात? हा पॉईंट परत सांगायला नको, एव्हरीवेअर यू आर स्टडिंग सेम थिंग, बरोबर ना? हॅलो.
हाँ सर.
हा. So
Yes, sir.
expected population is into 2050 it is 1.5 billion in India. We have to feed that much population. So it is very important to study new methods. Now I will tell a recent research. Hydroponics, you know, एक 6 महिन्यापूर्वीचा रिसर्च पब्लिश झालेला. I will discuss with you how they have increased the productivity. Hydroponics, you know, hydroponics.
नहीं
नाही
सर.
सर.
Hydroponics is what they will cultivate the plant in nutrient media without soil, with artificial or natural sunlight. Soil वापरायचीच नाही. So if you are not using soil, you will provide all the required nutrients in soluble form. बरोबर का?
हाँ सर.
So in soluble form you will provide these nutrients and the plant will be grown in control conditions. Now what are control conditions? You will provide that plant CO2. You will provide that exact amount of nutrients to that plant. You will provide exact amount of light day cycle, and the plant naturally it will get productivity will increase many times. So instead of getting 1 kg produce, you will get 10 kgs, and the crop duration is also reduced. आलं लक्षात हे?
हो सर.
मी तुम्हाला नेक्स्ट टाइम जर हे असेल स्पीड असेल तर तुम्हाला हायड्रोपॉनिक्सचा व्हिडिओ दाखवेन. सो इन व्हॉट हॅपन्स इन बिग सिटीज, if you consider, for example, now if you study the various vegetables, so what vegetables we required? Coriander, kothimir we required, methi is there, palak is there. So life cycle of all these vegetables, leafy vegetables, very small, बरोबर ना? किती दिवसात they mature in 1 to 2 months. We in our
हो सर.
Magdi we eat burgers. खाल्लाय सगळ्यांनी बर्गर? बर्गर खाल्लाय का?
हो सर.
हा. So in that burger the leafy part is present, it is called lettuce. बर्गर मध्ये जो ग्रीन कलरचा असतंय leafy part, that is lettuce, and this lettuce has got lot of nutrients and it has got lot of demand, and it will grow if proper conditions are given in hydroponics. It will come to harvest within 45 days, but outside of this, in natural environment, it will take 2 months or 2.5 months. So double the time period it will take outside, and in hydroponics we can harvest it in below 40 days. So what they're doing, and if you're producing artificial light, if you consider our biotechnology department, we are on second floor. If we go on putting artificial hydroponics, on every floor we can cultivate it, and on each floor we can 10 layer of plants we can fit in. So what is the productivity? It is tremendous. So in Japan, what they are doing, there is a software, lot of software companies are present in Japan. So when their production was shut down, semiconductors, so what they did, they used these spaces to cultivate this lettuce. मग एका स्मॉल एरियामध्ये वी कॅन इन्क्रीज द प्रोडक्टिव्हिटी 500 times. So what they did, a researcher in America, what they did, a group of researchers, they took wheat गहू घेतला त्यांनी, and they cultivated this in hydroponics, and they created a multi-layer farming system. So naturally they consider, if you consider this 1 hectare area, 1 hectare means 2.5 acre. तर त्यांनी काय केलं, 2.5 एकर मध्ये गहू कल्टिवेट केला नॅचरली. सो नॅचरली विल गेट प्रोडक्शन हाऊ मच, 200 to 300 kgs नॅचरली 2.5 एकरामध्ये. बरोबर बरोबर का? हॅलो,
हो सर.
3000 kgs, or we can calculate it, that is 2.5 to 5 metric tons. But they did this in hydroponics and they got a bumper crop of 700 tons, 700 metric tons. किती पट इफिशियन्सी वाढली? 300 times they could produce. and एरिया किती? म्हणजे instead of getting 350 or 400 hectares, they produce it in a single hectare. मग तुम्ही इमॅजिन करा की there is lot of pollution, no artificial sunlight is there, natural sunlight is not present, lot of smoke is there, and land is very less, water is also less. So what they did, they will construct this large hydroponics towers. So future मध्ये अशी कंडिशन आली तर now scientists are working in this direction. आलं लक्षात? हॅलो,
हा सर.
हा. बट हे सगळं आहे, आपल्याला दिसली प्रोडक्टिव्हिटी, पण सेकंड पार्ट पण काय बघायला पाहिजे आपल्याला? इकॉनॉमिक्स. इकॉनॉमिक कॉस्ट ऑफ cultivating or constructing these towers, circulating this artificial light, water, minerals, that is also very large. But what will happen, कालांतराने काय होईल, समजा फूड वाढत गेलं, समजा wheat we are getting now at 30 rupees, 40 rupees. If it goes for 3000, 5000, what they will do? They can cultivate in artificial systems. So people are working worldwide very hardly to produce this new method, because आता बघताय तुम्ही इकॉनॉमिक लॉसेस खूप झाले आपल्याकडं हे कोविड आल्यामुळे. सो व्हॉट हॅपन, द पीपल आर गेटिंग जॉबलेस. ड्यू टू दॅट व्हॉट विल हॅपन, दे कॅनॉट फीड देमसेल्फ. So what will happen? Theft and all these things, they will get on increasing. चोऱ्या खूप होणार, अनरेस्ट होईल पीपल मध्ये, इट विल गेट कम्युनल व्हायलन्स सगळ्या होतील. तरी सगळं स्टॉप करायचं. त्यामुळं ह्या सगळ्या सिस्टीममुळे काय होणार आहे, इट विल कॉज अ लॉट ऑफ अनरेस्ट. मग हे सगळं थांबायचं असेल तर यू हॅव टू वर्क व्हेरी हार्ड फॉर फूड सिक्युरिटी. And in this food security, which country has developed lot of techniques, agriculture productivity मध्ये वर्ल्ड मध्ये which country is highest research and development आणि application कुणी केले माहिती आहे? डेव्हलप्ड ऍग्रिकल्चर कुठल्या कंट्री मध्ये आहे माहिती आहे? कॅन एनीवन टेल?
इस्रायल.
yes, बरोबर, इस्रायल. If we consider Israel area, it has got only one river. That is also a conflict between two nations, and that river feeds to the Dead Sea. If you have heard of Dead Sea, lot of salinity is there. If you fall in Dead Sea, you cannot drown. Floating condition मध्ये राहणार, एवढी salinity Dead Sea मध्ये. And everything is desert. And area if you consider, our सातारा डिस्ट्रिक्ट, कोल्हापूर डिस्ट्रिक्ट आणि पुणे डिस्ट्रिक्ट, एवढे 3 डिस्ट्रिक्ट आहे की नाही, मिळून तेवढा एरिया फक्त असेल इस्रायलची लँड. बट दे आर प्रोड्युसिंग vegetables to whole Europe. This drip irrigation, greenhouse technology, everything was developed by Israelis. And adverse condition, if you are getting rainfall, ना महाराष्ट्रात जेवढा रेनफॉल येतो त्याच्या 5% only rainfall Israel gets, 5%. So this is the technological advance. Why? Because they invested lot in biotechnology and agriculture. Okay, next slide. Now, what are the benefits of agriculture biotechnology? I have explained. First is nutritional status. You have heard of golden rice? ऐकलं सगळ्यांनी?
हो सर.
हो सर.
Vitamin A supplemented. So सांगा नको
Yes.
separately. Then enable the crops to grow in hostile habitat. In hostile means there is, imagine there is desert, and the desert is having all rock surface. No, not a single gram of soil is there. But using this hydroponics, on that rock also you can cultivate plants in a tower. Third is tolerance to abiotic stress. Drought आहे, heat आहे. So if you want to do agriculture in Rajasthan or drier areas of Maharashtra, there also you can develop these crops and you can do the farming. Then designing plants with reduced water requirement. So reduced water कोणाला असतंय तर जे डेझर्ट प्लांट्स आहेत, झेरोफायटिक प्लांट्स, these are reduced water requirement plants. We can transfer the genes into our crop plants. Then fifth is increasing shelf life of the fruits and vegetables, flowers. नाव मी मागच्या कालच्या लेक्चरमध्ये सांगितलेलं की 30% ऑफ फूड प्रोडक्शन लॉसेस आर ड्यू टू less proper management, and shelf life is very less. If you consider leafy vegetables in rainy season, they will be fresh only for 3 to 4 days. If you consider this tomato, fruits like tomato and other fruits like beet or all these things, they will also remain outside only for 8 to 10 days. So if you increase the shelf life of these fruits vegetables, then naturally they can remain viable for more period. In tomato, the research is done on a large scale, and they have developed a fruit varieties which has shelf life of 1 month outside the fridge. Nothing will happen for the tomato. Then next is resistance to pest and diseases. आता पेस्ट अँड डिसीज साठी बायोटेक्नॉलॉजी एक्झाम्पल कुठलं आहे? बीटी कॉटन. बरोबर ना? बीटी कॉटन इज अ व्हेरी good example of biotechnology application in this pest and disease resistance. Then transgenic traits with the traits can be also used in modern agriculture. So in this way the modern agriculture is developing. Now molecular markers, if you come for molecular markers झाले तुम्हाला? Have you studied molecular markers?
Yes, sir.
So for example, I want a molecular, if I'm doing research on wheat and I've got thousands of wheat cultivars, say for example I've got 10,000 cultivars, then I want to develop a wheat variety which is short in length but has a growth or productivity very large. Wheatचं काय पाहिजे आपल्याला की it is drought resistance and it is short in length but the productivity is very high. So these characteristics will have 3 molecular markers: drought resistance, the gene for short dwarfness, and other is high productivity. Productivity high कशी मिळेल? Wheatचं जे वरचं पार्ट आहे, fruiting part, where the wheat is present. So number of wheat grain should be high. त्याची लेंथ हाय असली की तुम्हाला हाय प्रोडक्टिव्हिटी मिळणार. The length or weight of that wheat should be high. So what I will do, I will take these 3 molecular markers, and at a time what I will do, I will remove DNA from this 1000 wheat germplasm, and I will do only go on doing this electrophoresis and PCR. मग त्यातले काय मिळाले मला? आउट ऑफ दिस 1000 I will get 50 clones which are having these genes, and I will do breeding between these 50 clones. Instead of doing breeding in thousands, हे reduce कुठं केलं मी? फक्त 50 clones वरती आलो. So this is the significance of molecular marker. आलं लक्षात? हॅलो.
हा सर.
हो सर. हा सर.
घेतलं, आलं तुम्हाला लक्षात? सेम अप्लाइज फॉर अदर क्रॉप्स. What you have to do, you have to isolate DNA. Then what you will do, you will have got 3 markers for dwarfness, drought resistance, and increase yield. And you will run this with gel electrophoresis. Gel electrophoresis वरती आल्यानंतर तुम्हाला बँडिंग मिळेल. What you will do, this banding you will take photo, and the banding वरनं you will design a database. In that naturally this will get characterized. Bioinformatics tool वापरलं तुम्ही, आणि टूल वापरून काय केलं तुम्ही? Elite germplasm separate केले. How much your time was saved? 10 वर्ष लागणार. That research was, we can do it in 2 years, 3 years. Second is varietal identification and clone fidelity testing. Now if you are doing टिश्यू कल्चर करतात माहिती तुम्हाला ह्याचं? बनाना टिश्यू कल्चर सगळ्यांना माहिती ना? बनाना टिश्यू कल्चर.
हो सर.
In this banana tissue culture, what happens? They go on subculturing. They will take initially one rhizome and they will go on subculturing to produce lot of plants. मग आपण समजा 10-10 वर्ष 20-20 वर्ष सबकल्चर, इफ यू कीप ऑन सबकल्चरिंग, विल गेट लॉट ऑफ प्लांट्स. But tissue culture system is a artificial system, and there are so many variations. And if these variations are negative, means for example you are doing tissue culture for banana, and the productivity of the tissue culture raised plants is very less. For example, the size of this banana is very less is as compared to mother plant. ते प्लांट्स काय कामाचे आहेत का? नाही. त्यामध्ये व्हेरिएशन झालं. सो दिस क्लोनिंग फिडिलिटी टेस्टिंग कसे करणार? तर ॲट एव्हरी स्टेप यू विल रिमूव्ह some DNA, some plants, and you'll isolate the DNA. And with that DNA, you will learn these molecular markers with the parent DNA. मग त्याच्यावर तुम्हाला कळेल की कुठं मार्करमध्ये डिफरन्शिएशन झालंय का. आलं लक्षात?
हो सर.
हो सर.
काय केलं त्यांनी? फॉर एक्झाम्पल आय टूक वन बनाना प्लांट, अँड आफ्टर दॅट बनाना प्लांट आय हॅव सबकल्चर्ड इट. सबकल्चर केल्यानंतर दिस इज फर्स्ट जनरेशन. एका प्लांटचे मला किती मिळाले? 4 प्लांट्स. From this 4 plant I did cut and I put again in 4 different bottles. मग तिथं किती मिळाले मला? 4 bottles मिळाले. Again these 4 plants developed 10 plants. म्हणजे every bottle I will get 40 plants total. Again what I will do, from these 4 bottles I will produce 16 bottles. त्यानंतर 64 bottles. ही झाली प्रत्येक स्टेज झाली. फर्स्ट, सेकंड, थर्ड, फोर्थ, सब कल्चर झालं. अशी मी कंटिन्युअस किती वेळ करू शकतो? जोपर्यंत त्याची फिडिलिटी त्या पॅरेंट बनाना प्लांट बरोबर मॅच होती. ती जर चेंज व्हायला लागली तर काय होणार? आय विल गेट प्रोडक्टिव्ह लॉसेस. ते पॉझिटिव्ह असेल किंवा निगेटिव्ह असेल, पण ती रिस्क घ्यायची नाही. तर हे डिसाईड करण्यासाठी आपण मॉलिक्युलर मार्कर्स वापरतो. देन असेसमेंट ऑफ जेनेटिक डायव्हर्सिटी. आता डायव्हर्सिटी, इफ यू आर स्टडिंग दिस व्हीट, आपल्या ह्याच्यामध्ये किती किती क्लोन्स आहेत व्हीटचे, त्याच्यामध्ये किती जेनेटिक डायव्हर्सिटी, ते तुम्ही मॉलिक्युलर मार्कर्सने बघू शकता. देन व्हॅलिडेशन ऑफ जेनेटिक रिलेशनशिप. आर दिस प्लांट जेनेटिकली रिलेटेड? देन मार्कर असिस्टेड सिलेक्शन. मी पहिलं एक्झाम्पल दिलं व्हीटचं, हाऊ दे हॅव यूज दिस मार्कर असिस्टेड सिलेक्शन. देन देर आर मार्कर सिस्टम्स, आयसोझाईम मार्कर्स आहेत, डीएनए बेस्ड मार्कर्स आहेत. सो इन मार्कर्स मागच्या याच्यामध्ये तुम्हाला डीपमध्ये झालेलं आहे. झालेलं ना मार्कर्स हे सगळे?
हो सर.
हां, ते ऍप्लिकेशन सांगा नको. तो दिस मॉलिक्युलर मार्कर्स वाय हॅव टू, बिकॉज वेन databases, there you can know what gene it is, what character it will express, what enzyme it will produce, what protein it will produce. तर ह्या सगळे तुम्हाला गोष्टी कळतील कधी? तर वेन यू कम्पेअर इट, वेन यू यूज दिस बायोइन्फॉर्मॅटिक्स टूल. तर ह्या सगळा जो डाटा प्रोसेस करण्यासाठी बायोइन्फॉर्मॅटिक्सची गरज लागली. त्या सो द scientists they have developed these different bioinformatics tools. So molecular markers are one of the reason lot of research has took place in this bioinformatics. Then next is steps in transgenic. How they are producing transgenic animal, plant, bacteria. First what they will do, they will identify the genes. Then they will prepare this forward reverse primer. They will isolate that gene. They will put that gene into a cloning vector. मी एक्झाम्पल दिलं तुम्हाला, क्लोनिंग वेक्टर. मागचं सगळं आपल्या जीचं ते वेक्टर्स वरतीच होतं. तर त्या क्लोनिंग वेक्टरमध्ये त्यांनी तो ट्रान्सफर केला जीन. देन दिस जीन इज ट्रान्सफर्ड इनटू ॲग्रोबॅक्टेरियम. देन फ्रॉम ॲग्रोबॅक्टेरियम इट इज ट्रान्सफर्ड टू अ प्लांट. आता प्लांट मिळाल्यानंतर तुम्हाला टिश्यू कल्चरमध्ये यू गेट हंड्रेड्स ऑफ प्लांट्स. आउट ऑफ दॅट विच इज द ट्रान्सफॉर्म प्लांट विच इज हॅव्हिंग स्टेबल जीन. स्टेबल जीन म्हणजे वेन वी ग्रो दॅट प्लांट अँड इफ द वी हार्वेस्ट द सीड्स अँड वेन वी कल्टिवेट दॅट सीड्स, द जीन शुड बी ट्रान्सफर टू द सीड्स. एकाच जनरेशनला येत असेल then that is of no use. So selection and regeneration of the transgenic plant. Then transgene stabilization, whether the gene is stabilized. How you will know the gene is stabilized? You will go for expression, RT-PCR you will do. Whatever that gene is producing any protein or enzyme, you will separate the protein and you will do protein identification. If it is producing toxic plant, for example, if you are transferring BT, it will produce cry protein. So you will isolate the cry protein whether it is present in that plant or not. From that you will know the stable expression of that gene. Then you will take trials and then you will release a variety. So these are steps in transgenic plants development. तसं ह्या वेगळ्या वेगळ्या स्टेपला तुम्हाला वेगवेगळे बायोइन्फॉर्मेटिक्स टूल वापरून करून वी कॅन ईज द प्रोसेस. ठीक आहे, थांबायचं का? हॅलो हॅलो. आणि आज एवढी कमी काय आहेत, फक्त 8च जण. हॅलो.
रोज 10 ते म्हणजे 10 पर्यंतच लेक्चर सगळे 9 ते 10, 10 ते 11 पर्यंतच असतात ना, त्याच्यामुळं कुणी बघितलं नसेल मेसेज तुमचा.
बरं ठीक आहे, चालेल. मग कोणाला काय डाऊट आहे का, विचारायचं का?
नाही सर.
आवाज आज सगळ्यांना येतोय? एव्हरीवन कॅन हिअर प्रॉपरली?
हो सर.
हो सर.
हो.
चालेल, ठीक आहे. आणि